Okay, the Myxomycota today. This is one of two groups, the Myxomycota and the Mycomycota, that are in this group of slime molds. We'll see in the phylogeny in just a minute where they fall. They are not in the same monophyletic group with the other fungi. So we've passed out of our mycelial fungi now. We're no longer dealing with the mycelial fungi, and we're dealing with these groups that are weird in a lot of different ways, even some ways weirder than the regular fungi, and these are the plasmodial fungi. So now there's differences between the Myxomycota and the Mycomycota in the form of the Plasmodium. You don't know what a Plasmodium is yet, but the Myxomycota have a true Plasmodium and the Mycomycota have a pseudo or false. And so what is a Plasmodium? Well, you know the roots, plasm, something formed, and IUM little, not so little sometimes. Doesn't tell you really much here. Knowing the roots in this case is not very informative. A Plasmodium, a true Plasmodium, is a multinucleate mass of protoplasm. With no internal cell walls. I told you we'd be doing the fungus that ate Tokyo today. So I'm sure there is a Japanese horror movie that involves a Plasmodium swallowing a massive protoplasm, swallowing Tokyo. Took over right after Godzilla came through. The pseudoplasmodium is cellular. Yeah, so why don't you, it's wonder why we would call this a Plasmodium, but it's a cell, I don't know how to describe it yet. Cellular mass of cells, this isn't really right. Because it has both unicellular and, in quotes, multicellular. Make sure you've got quotes around that, stages, because the multicellular stage is not really completely multicellular. We'll come to that next. We'll do the Mycomycota next. So this brings us to my favorite root of the whole semester. Did you all look up MYX and know what MYX means? Because it is perfect for prepubescent boys. It is the word that was made for them. Nasal discharge. It is the nasal discharge fungi. I can tell none of you have been prepubescent boys in this room. So it's also can mean slime, but nasal discharge is the best. For this. So these are the nasal discharge fungi, and they look exactly like that, almost exactly like that. Here they are, the slime molds. This is both the Mycomycota and the Myxomycota in that group. So you see it is not a monophyletic group with the fungi. It's really only a monophyletic group with itself. We had to make one cut. This is the only group we could remove with that one cut. And so that's caused some people to suggest it should be its own, perhaps even its own kingdom. Although I was looking at a nice website that talked about the Myxomycota yesterday, and they were saying, well, you know, it's really more related phylogenetically over here to the animals, but we're going to put it in. Where do you put anything you don't know where it goes? Remember from the very beginning? If you don't know where it goes, put it in the protista. You put it in the protista, that grab bag kingdom that has a bunch of unrelated organisms in it. We've split it up for this class. And so that website, and you would see many others, will stick this thing in the protista. Because why not? Here it is. Today, this Petri dish. Tomorrow, Tokyo. Day after tomorrow, the world. There it is. This is the Plasmodium. Here it is as a close-up. This stuff here, that's the Plasmodium. This is an oat. So that's a rolled oat. And it's growing on the rolled oats, so that structure to the organism that you see there is the oats underneath it. This organism is growing on top of it. Here you see that close up as a through a thing. There's the Plasmodium. See, it does have some internal structure. There is some structure there, but it is a mass of protoplasm surrounded by a cell membrane, and it's got a gel-like consistency on the outside of it, but there's no cell wall there, and there are no internal cell divisions. So you'll see it in lab growing something like this. We'll have some Petri dishes that look something like this. You can see there was a spore or an initial cell that was inoculated there. And there's a wave front now growing in all directions, but more in this direction. So there can be directional growth to this thing. And here is some of that. If we were to look in this region here, this ropey mass would look like that. We'll have a bunch more pictures of that. You see, but that is not the main wave front. That's not the main part of the organism that's growing toward the east, actually. Tokyo's east from here. Actually, so far it's probably east or west. Why does that have an annotation on it? That's interesting. Well, there's the Plasmodium. Let me get some white. This area then is the Plasmodium. It's got that ropey structure to it, so it looks like it's hyphal, but it's not. There's no hyphae there. This is a fruiting structure. I'm going to learn a bunch of names for this fruiting structure in a minute. It's going to produce, or all the fruiting structures are going to produce, haploid spores. So they're going to produce haploid spores. Now, this species of Plasmodial slime mold is not growing in the United States. It's actually growing in Austria. I was in Austria a few years ago for a scientific meeting, and I had an opportunity with a field trip from the meeting to go in some really beautifully undisturbed areas of some reserves. A reserve that was from a very rich family. If you don't know, if you perhaps have heard of the Rothschilds, who were very important financiers of Europe in the Middle Ages and on till now, and they had huge, huge areas that they reserved for their private hunting lodges, and that some of that has been given over to the state now. And this was one of the old Rothschild reserves, very limited access. We could go in there, saw beautiful plants, all kinds of things, and this fungus was growing in there. This is the, what would you name that fungus? If you saw that out there, what does that look like to you? If you were, now really, you just have to put yourself in that prepubescent boys. I know that's really hard for all of you, especially girls, but put your in that mindset there, and you look at that thing and you say, you all have dogs? Yeah, it's that. What is it? It's a dog vomit fungus. This is the dog vomit fungus. Has no internal structure in that thing, except it's going to produce a whole bunch of spores there. I'm not kidding about its name. That really is the English name for that species, the common name. Somehow I opened the one with notations on it, which I wasn't supposed to do, but anyway, we got some notations on this, and you can see that the slime mold is moving in that way. So that front is here. And this is all Plasmodium. All this back here is Plasmodium with that ropey mass. It's actually in this place here between these humped up portions. There it is. Here it is again. That's the piece there. That's that plasmodial part that was like what we saw on the last page. In this case, it's forming the fruiting structure. So it's also down in here, it's also growing inside that log. It's growing inside and on the surface of that log. Here it is, what's happening in there. I wish we could see this in lab, but we've never seen cytoplasmic streaming like that. I've got to play it a few times here for you, but you can see this is sped up. But you can see there's the cytoplasm moving inside the plasmodium toward that front, and there is no internal structure there at all. It just moves out into that mass in the direction of movement, following the wave front. And there it is again. Direction of movement would be this way. These organisms are live by phagocytosis. Phagocytotic. So they engulf bacteria. So you know that from the other fungi, they engage in extracellular digestion. They secrete enzymes outside of their body and digest the organisms extracellularly, and then take the materials that they've digested in liquid form into the mycelium. The members of this group, of the Myxomycota and Acrasiomycota, don't do that. They engulf, they're scavengers, and they will engulf bacteria and other small organisms. So they live by phagocytosis. Here's the Plasmodium in a section. You know, I wish I could show you some cell walls or some internal structure. There's not much you can see. These are vacuoles, I'm sure, that are stained here red. The nuclei should be staining a little dark. There's probably some of these dark spaces here are nuclei. There's no organized special structure there. In fact, if there's any little bit of structure we see, these areas here, you can see that white there, those are probably artifacts of the fixation process that allowed the scientists to prepare this section. So at some places in its life cycle, the Plasmodium will dry out. If the conditions worsen, loses moisture which it needs for growth, the Plasmodium will dry out and enter a resting stage. And the Sclerotium is the resting stage. The Sclerotium can have many different forms. This is just one of them in one species. You can see it looks like it's kind of shriveled up, and there's actually some little spore cases there probably too, but mainly we're looking at this hard resting stage 4. Did we do the root for sclero already? Means hard. And otium would be, TIUM would be little. It's not so little in this case again, but probably it's used, that root is probably used because Plasmodium and Sclerotium are similar. So Sclerotium, it gives us also medical terms like scleroderma. It's a very serious disease of the hardening of the connective tissue. It's a genetic disease. And I mention it because my uncle died of complications of scleroderma. It was technically from kidney failure, I believe, but it was brought on by scleroderma, which is a very, very serious disease. But again, the same root. Here we have a section through the sclerotium, and now we can see the nuclei. And those are not cell walls. Those little white areas, those are not cell walls. That's just, again, kind of an artifact of how this is prepared. So no internal structure of these Plasmodium or Sclerotium. Here's the fruiting structures. So they can have lots of different forms. And they're given a number of different names because of that. These are the general names for them. The Plasmodiocarp, and you know the roots for Plasmodiocarp, plasm, molded or formed, carp, fruiting body. So that's pretty descriptive now in this case. It's just some little part of the organism, or the whole organism, that's formed up into a fruiting body, something that's formed a fruit of this. Ethalium, ethal means smoke. So I had a question the other day from a student about roots and which ones you're likely to see. On exams, you are likely to see these. You are not likely to see that one, because this is the only word that the root ethal occurs in. Ethalium would be little smoke. It's called little smoke because when these things break open, many spores are shed. Just imagine that's dog vomit slime mold opening up. It would just produce, like a member of the Gastromycetes, produce masses and masses of spores. So little smokes. Here's another kind of Plasmodiocarp. This one looks like a sporangium, so we can in fact call this one a sporangium. You'll see these in lab, but unfortunately the ones in lab we have are not, the spores have been shed already. And when they've been shed, what you notice is that there are structures through here, little branches that come out like this. You can kind of see them a little bit in here. And those branches have been preserved in the material we have in lab, but all the spores have been shed. So you'll see the structure that supported the spores, and there is a covering here on this, which makes it the box, the angium. And that'll be next week to look at that. So there's sometimes we could say there are the Plasmodiocarps have a form of a sporangium. But here's one that doesn't. Here's a Plasmodiocarp, looks nothing like a sporangium. Here's the Plasmodium over here. And the Plasmodiocarp. Black doesn't work very well. We need to get that light replaced. It is not serving us very well. And here it is, cut open. So these are the spores. And in the Plasmodiocarp, they are haploid. So just a mass of haploid spores. Let's put this together in the life cycle and see what we've been looking at. We haven't seen all parts of the life cycle, but we've seen many parts of it. So when we want to find the life cycle, we want to find meiosis and syngamy, and then they've broken syngamy into plasmogamy and karyogamy here, but there's no reason we have to do that in this organism. Plasmogamy and karyogamy take place right after each other, as in normal, so to speak, organisms. So we can write meiosis there and syngamy here. So that gives us our diploid and haploid portions of the life cycle. So it's a normal dibionic life cycle with the provision that there's something really weird over here, this Plasmodium. And over on this side, we haven't discussed yet, but it's going to be unicellular. The organism is going to be unicellular in the haploid stage. Here's our resting stage, our Sclerotium. There's the Sclerotium, so it can undergo the Plasmodium. Which is the main assimilative stage. Assimilation means to grow, to take in nutrients and grow. assimilative stage of the organism, it can enter this resting stage then in bad times. The sporangium, or the Plasmodiocarp, forms, and initially the cells are diploid, but within the Plasmodiocarp they become haploid. So for us, we're just going to consider the Plasmodiocarp to be haploid. It's like, all sporangia are like that. They go through a stage when they're initially formed and they're diploid, and then meiosis takes place. And we've been considering them to contain the haploid spores. And so here we have the haploid. The spores are going to germinate, and they produce these unicellular organisms. So there are amoebae. Now there's the amoeba, they're not the amoeba of the protista, but they are the amoebae of this organism. Or there are these flagellated cells. And these two forms, these two unicellular forms, are interconvertible. So if we were growing these in culture, we can change the conditions just slightly. We can get amoebas at one point, and they can switch over and produce the flagella, change the conditions back. The flagella will be retracted or reabsorbed into the organism, and amoebas will be produced. So they're interchangeable in that during the life cycle of the organism. Either of them can function as gametes. So there's a plus gamete and a minus gamete there, and they undergo syngamy. So it's a minor assimilative stage. They can live on their own to some extent, but the main assimilative stage is the plasmodium. These guys mainly are functioning there as gametes. They're living long enough to fuse as gametes. So the haploid stage is not long-lived. So in other words, you could talk about this as a monobiotic diploid life cycle too, if you considered those just to be gametes on the amoeba side. And that's supposed to be a So here's a nice, I hope, little video. So this is going to show, I'll turn off the lights. It's going to show the growth of the Plasmodium first. The sound is really annoying, so I'm not going to plug it in. You might hear it a little bit from my computer up here. At some point, it's going to switch over and show Myxomycota and growth of the mushrooms, and I'll stop it then again and show you that. But for right now, this is Myxomycota. Sorry, it's such poor resolution. But there's the Plasmodiocarp forming. There's the front of that. There it is, really nicely growing out. Even better picture of the Plasmodium growing, heading for Japan. And there's the Plasmodiocarp forming, almost sporangial-like in those cases. Here's mushrooms. Okay, so now we switched over. They just didn't distinguish here between the Myxomycota and the Myxomycota, but this is clearly Myxomycota. We're getting the mushrooms formed. You can see here's the remnants of the universal male on this mushroom. There's that little button stage. So there'll be a number of mushrooms that we'll watch now. Again, it's time lapse. And they would be shedding spores all through that last stage there. This is one of the stinkhorns, and it's not fully formed now. The really cool stuff starts happening. The basidia are on this. So there's no gills, but It's replaced by that net-like structure that comes out of underneath the calf, and that's covered with Plasmodium. Pretty amazing. I think this is going to be the same thing again from from farther out, so you see it again. And just the very ephemeral nature of these mushrooms. They don't last very long. One of the shelf fungi, growing as a wood rot, composing that log. And shedding the spores during this period of time when you see those caps moving around. Okay, that's it.
That's it.